The next speaker is Xin-Yi Chang from National Taiwan University, and she's going to tell us about the development of a butanol-tolerant microorganism.
Good evening, ladies and gentlemen. Okay, I'm Xin Zhong, come from National Taiwan University. My study was about to development of one butanol-tolerant microorganism. As a primary source to run the world's industries and to provide us a necessity of lives, the end of petroleum is a foreseeable reality. From this figure, we can see that this is the oil prices from 2000. Today, the oil prices is almost four-fourths compared to the 8 years ago. So people say that the thing is value when it is rare. So what can we do and how can we do if the oil runs out? I think the alternative energy might be the good question for a good answer for these questions. Okay. And this table compares several alcohols to gasoline that we can use as the alternative energy. Now, as we can see that butanol has higher energy content compared to methanol and ethanol, and it is almost the same as gasoline. And the octet number of butanol, however, is lower than these two alcohols, but 96 octet number is not too bad for the cars. The vapor rate, the vapor pressure is low, and it means that it is healthier to us and it is healthier to us. And okay, the solubility in water is about 7%, and it means that it is easy to separate from water with less energy cost. Okay, let me introduce you the 1-butanol production history. Traditionally, 1-butanol is produced using acetyl-butanol ethanol fermentation by Clostridium, and it is one of the oldest known industrial fermentation. However, the actual fermentation is so complicated, so almost all butanol is produced via petrochemical routes. The production of butanol was limited by severe product inhibition, and it can be inhibited by 1% butanol, and the butanol concentration in conventional ABE fermentation is usually lower than 1.3%. So what we are going to do is try to find some user-friendly microorganism and to use it for producing butanol. And first, we have tried to make it tolerant to the product. So, okay, this is what we are using. We are using Escherichia coli as a model to it is a user-friendly microorganism, and it has been well characterized, and it is suitable for produce valuable metabolite. And the high growth rate is the E. coli grow faster than acetylbutylicum. In our major goal, we are trying to screen out one butanol tolerant strain from screening from genome-wide single-gene deletion mutants, and to find some 1-butanol tolerant protective proteins using proteomics approaches, and to develop 1-butanol tolerant E. coli use the result from these two parts. Okay. There is an experimental design. In the first two parts, we are going to find the mutant as a candidate and find some protective proteins and combine them at the third part. In the first part, we are going to we screen out from 3,985 single-gene mutants, also known as KL collection, which was constructed by Dr. Maury, and we screen it with 2% butanol, and finally we found some mutants that tolerate to 1-butanol. This is the butanol tolerance of wild-type E. coli. As we can see that most concentration higher than 1.5% exhibit severe inhibition on E. coli growth rate. So we choose 2% as the criteria to screen the KL collection. From this 3,985 single-gene deletion mutants, we screen out 85 mutants has higher butanol tolerance compared to the wild type, and the 13 strands higher than 20% to wild type was selected out and for further confirmation. And the first 4 were characterized by phenotype microarray biolog analysis. By the analysis, we found that all of these 4 mutants exhibit high resistance to osmolality challenge as well as multiple antibiotics. And we double-check the result. At the final, we choose these 3 strands as the host cells for the following experiment. And the second one, we're trying to find some 1-butanol related proteins using proteomics approaches. We're using 2-dimensional electrophoresis to find out some protein expressed differentially in butanol-treated condition. And in this result, we can find after butanol treatment in wild-type E. coli, we find some protein upregulated in this condition. They include some antioxidative proteins and ATP transporter proteins and some membrane transporter protein and the SOD, something like that. And the protein involved in glycolysis and amino acid degradation as well as the membrane signaling was downregulated in this result. From this result, we choose all of the upregulated protein and express them in wild-type E. coli and see if it is contribute to 1-butanol tolerance. As at these 13 strands, we found there are 6 strands exhibit high tolerance to 1-butanol. And then we are going to use the 6 proteins that we identified in the second part into the 3 candidates that we screen out from the first part and generate the JH strands. In the JH strands, we test the butanol tolerance on each one and find there are 3 strands showed high tolerance to 1 butanol. Also, we test how much butanol they can tolerate. We found that in these 3 strands, they can tolerate 2.5% to 5% of 1 butanol and 3.5% to isobutanol. Also, there are 1 strand can tolerate in 1-propanol and isopropanol, different kinds of alcohols, but they can tolerate to that. Compared to the control strands, which is tolerate to 2% of propanols only. In the summary, using our approach, we can we obtained 3 cal strands and use it as the candidate host cells. And by proteomics approach, we find some proteins involved in butanol tolerant process, and it might be protection of DNA and membranes and against oxidative stress, as well as survival for extreme environmental conditions. For the combination strategy, we can obtain 3 JH strands which display high tolerance to 1 butanol, 1 isobutanol, and 1 strand tolerate to propanols. So we think the 3 JH strand might be an ideal model to applying on alcohol production in the future. Okay, and I want to thank my advisor, Dr. Xuefeng Ran, which is come from National Taiwan University, and our collaborator, Xuancheng Huang from National Yangming University, Dr. Hirotada Mori from NIST Japan, and Dr. Shuitian Chen from Seneca, Taiwan, and Dr. James Lau from UCLA. And this is our member in our laboratory, and thank you for your attention.
Thanks, Xin-Yi. So now the floor is open. Any question? One point I may have missed. You mentioned that at the end you've worked with the 3 mutants. Have you confirmed and mapped the mutation to be at those genes, or are you simply based on the proteomic data?
No, that's the screening part.
Right.
You mean a screening part?
Right.
And we only test on what kind of phenotype they exhibit.
Right.
Yeah.
So you have not confirmed what mutations they are.
Yeah.
Okay.
I had a similar question. So.
Okay. Question at the back.
What are the advantages, do you think, of doing an approach this way as opposed to, say, just random mutagenesis and selection by exposing them to high levels of butanol and stuff?
I think the evolutionary metagenesis is taking a longer time, and we think our approach might have some impact on the time saving, but not always can be successful. Yeah, because it only mutate 1 gene at the same time. So we don't know what happened in the evolutionary strategy. So.
Now, may I have another question? Is that now that you have some of the mutants which are more tolerant, are you considering using those tolerant mutants and continue to go for a second round of mutation to boost up the tolerance?
Actually, we are undergoing this part. Yeah.
One more question.
I was just curious if you've tested these strains with other alcohols is the tolerance highly specific to 1-butanol, or is it actually a more general tolerance?
Actually, the strain JH L16, the last one strain, which can tolerate to 1-propanol, it is also can tolerate to ethanol, about 14% ethanol. Yeah. But other strains can only tolerate to 1-butanol only. Yes. Thank you.
All right. Do we have more questions? If we don't, let's thank all the speakers for this section for their excellent talks.